Come to biology, the scientific study of life. Responses to light are critical for plant success. Blue light photoreceptors and phytochromes as photoreceptors. Notice this inquiry: what wavelengths stimulate phototropic bending toward light? Experiment researchers exposed maize, coleoptils to violet, blue, green, yellow, orange, and red light to test which wavelengths stimulate to phototropic bending toward light. Results: the graph below shows phototropic effectiveness curvature perform relative to effectiveness of light with a wavelength of 436 nanometer. The photo collages show coleoptils before and after 90 minutes. Exposure to side lighting of the indicated colors pronounced curvature occurred only with wavelengths below 500 nanometer and was greatest with blue light. Notice the experiment and the conclusion. The phototropic bending toward light is caused by a photoreceptor that is sensitive to blue and violet light, particularly blue light as shown here. Notice that. Another inquiry: how does the order of red and far red illumination affect seed germination? Experiment. During the 1930, USDA scientists briefly exposed batches of lettuce seeds to red light or far red light to test the effects on germination. After the light exposure, the seeds were placed in the dark, and the results were compared with control seeds that were not exposed to light. Results: the bar below each photo indicates the sequence of red. Notice that of red light exposure, far red light exposure, and darkness. The germination rate increased greatly in groups of seeds that were last exposed to red light left. Germination was inhibited in groups of seeds that were last exposed to far red light right. Notice the conclusion: red light stimulates germination and far red light inhibited germination. The final exposure was the determining factor. The effects of red and far red light were reversible as shown here. Notice that. Another experiment or notice also another figure express or represents structure of phytochrome. Chrome 4, notice that, and chrome 4, these are. A phytochrome consists of two identical proteins joined to form one functional molecule. Each of these proteins has two domains. Notice also photoreceptor activity. One domain which functions as photoreceptor is covalently bonded to a non-protein pigment or chromophore. Notice also kinase activity. The other domain has protein kinase activity. The photoreceptor domains interact with the kinase domains to link light reception to cellular responses triggered by the kinase. Another figure, notice that represent also the responses or response seed germination control of flowering. Notice also synthesis, pure, slow conservation or conversion in darkness. Some plants, red light, far red light, and enzymatic destruction. Notice P if R responses seed germination control of flowering. Notice that this figure represents or shows the sleep movements of bean plants. The movements of or are caused by reversible changes in the turgor pressure of cells in opposing sides of the bulbini motor organs of the leaf. Notice noon and midnight and afternoon. Another experiment inquiry, how does interrupting the dark period with a brief exposure to light affect the flowering? Experiment. During the 1940, researchers conducted experiments in which periods of darkness were interrupted with brief exposure to light to test how the light and dark portions of photo period affected flowering in short day and long day plants. Result, notice darkness, flash of light, a critical dark period. Uh, Notice also short day plants flowered only if a period of continuous darkness was longer than a critical dark period for that particular species. 13 hours in this example. A period of darkness can be ended by a brief exposure to light. Notice B, long day plants flowered only if continuous a period of continuous darkness was shorter than a critical dark period for that particular species. 13 hours in this example. Conclusion. These experiments indicated that flowering of each species was determined by a critical period of darkness, a critical night length for that species, not by specific period of light. Therefore, short day plants are more properly called long night plants, and long day plants are really short night plants. Another inquiry is photochrome is or the pigment that measures the interruption of dark periods in photoperiodic response. Experiment. A unique characteristic of photochrome is reversibility in response to red and far red light. To test whether phytochrome is the pigment measuring interruption of dark periods, researchers observed how flashes of red light and far red light affected flowering in short day and long day plants. Result. A short day, long night plant, notice also hours, and notice a critical dark period. Conclusion. A flash of red light shortened the dark period. A subsequent flash of far red light cancelled the red light's effect. If a red flash followed a far red flash, the effect of the far red light was cancelled. This reversibility indicated that it is phytochrome that measures the interruption of dark periods. Another require if or is there a flowering hormone? Experiment. To test whether there is a flowering hormone, researchers conducted an experiment in which a plant that had been induced to flower by photoperiod was grafted to a plant that had been induced. Result. A plant subjected to photoperiod that induces flowering and a plant subjected to photoperiod that does not induce flowering. Notice graft. Notice also time several weeks, and notice the conclusion. Both plants flowered, indicating the transmission of a flower-inducing substance. In some cases, the transmission worked even if one was a short-day plant and the other was a long-day plant. Notice that.